Hey guys, welcome to another episode of the Human AI podcast. Today we're talking about metabolism. I'm here with Grok, an AI. It's going to be explaining these concepts to us. Please take everything that it says with a pinch of salt. And yeah, I hope you find this video informative and enjoyable. Having said that, Grok, let's continue. This is part 2, by the way, guys. So I should have said from the beginning. So if you haven't watched part 1, it's just click on the episode below in the channel, and it's going to make a lot more sense because we're continuing from there on. Okay, Grok. We left off talking about DHAP and G3P. So we have these two from fructose biphosphate, 1,6-biphosphate. And I want to ask you about what happens after to G3P. So, we've got G3P, the only one that continues through glycolysis, and tell us why it's the only one. And then GAPDH will turn it to 1,3-BPG, 1,3-biphosphoglycerate. And then for this to happen, NAD plus will become NADH. Then we have PGK turning 1,3-BPG to 3-phosphoglycerate. And for this to happen, ADP goes to ATP this time. Explain this to us.
All right, welcome back to part 2, diving right into glycolysis where we left off. So, we've got fructose 1,6-biphosphate split into two 3-carbon molecules, dihydroxyacetone phosphate, DHAP, and glyceralpha 3-phosphate, G3P. Only G3P continues directly through glycolysis because it's the molecule that the next enzymes are designed to handle. DHAP doesn't just sit there though, it gets converted into G3P by an enzyme called triose phosphate isomerase. This happens because the glycolysis pathway is set up to process G3P, so DHAP is quickly transformed to keep the flow going, essentially doubling the G3P molecules for the next steps. Now, onto G3P. The enzyme glyceralpha 3-phosphate dehydrogenase, or GAPDH, takes G3P and turns it into 1,3-BPG. Here's how it goes. GAPDH uses NADH as a coenzyme, grabbing electrons from G3P in an oxidation reaction, which turns NADH and adds a phosphate group to G3P, forming 1,3-BPG. This step is key because it's where glycolysis starts generating energy carriers like NADH, which will later feed into the electron transport chain for more ATP. Next, 1,3-BPG is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase, or PGK. In this step, the high-energy phosphate group on 1,3-BPG is transferred to ADP, forming APOT. This is the first payoff in glycolysis, where you start getting energy back after the ATP you invested earlier. The name PGK fits because it's a kinase, transfers phosphate acting on a glycerate molecule, and the reaction is efficient due to 1,3-BPG's high-energy bond, which makes the phosphate transfer to ADP favorable. So, in these steps, you're harvesting energy as NADH and APOT while moving the molecule closer to pyruvate, the end goal of glycolysis.
This makes a lot of sense. This makes a lot of sense in the way you explained it. Okay. And why is PGK and GAPDH the right enzymes? Are they enzymes? Okay. Then we have mutase making 3-phosphoglycerate to 2-phosphoglycerate. So explain that, how mutase is the ideal thing and how its name relates to its structure and function. Then tell me about how 2-phosphoglycerate becomes phosphoenol pyruvate with enolase. And lastly, let's talk about how phosphoenol pyruvate will become pyruvane with PK, with ADP turning to ATP, and why these steps are called SLP steps. Also, each reaction is occurred twice for every glucose molecule. I assume this is because you said, like you said, DHAP will become G3P in the end. Can you tell me the answer?
Let's keep rolling with glycolysis, picking up where we left off with G3P and diving into your questions about the enzymes, their names, and the final steps, including why these reactions happen twice per glucose and why some are called SLP steps. First, why are GAPDH and PGK the right enzymes? Glyceralpha 3-phosphate dehydrogenase, GAPDH, is perfect for turning G3P into 1,3-BPG because its structure has an active site that binds G3P and NADH tightly, allowing it to oxidize G3P while adding a phosphate from inorganic phosphate in the cytosol. The dehydrogenase in its name reflects its role in removing hydrogen, electrons, and a proton from G3P, reducing NADH to NADH. Its specificity for glyceralpha 3-phosphate is why it's named that way. It's tailored to this substrate. Phosphoglycerate kinase, PGK, is next, converting 1,3-BPG to 3-phosphoglycerate. PGK's structure is designed to bind the high-energy 1,3-BPG and ADP, transferring the phosphate group to make APOD-T. The kinase in its name indicates it transfers a phosphate, and phosphoglycerate points to its substrate. Both enzymes are ideal because their active sites are shaped to fit their specific molecules, ensuring efficient, precise reactions. Next, 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. Why mutase? Its name comes from mutate, meaning it rearranges the molecule, in this case, shifting the phosphate group from carbon 3 to carbon 2. The enzyme structure has a pocket that binds 3-phosphoglycerate and temporarily holds a phosphate group, allowing it to reposition the phosphate. This subtle change preps the molecule for the next step by making it less stable and more reactive. Then, 2-phosphoglycerate becomes phosphoenolate pyrate, PEP, via enolase. Enolase's name reflects its function. It creates an enol group by removing a water molecule, forming a high-energy double bond in PEP. Its structure is designed to bind 2-phosphoglycerate and catalyze dehydration, making PEP highly reactive due to its enol phosphate structure. This sets up the final energy payoff. Finally, pyruvate kinase, PK, converts PEP to pyruvate, turning ADP into ATP. PK's name is spot on. Kinase means it transfers a phosphate from PEP to ADP, and pyruvate indicates its product. PK's active site grips PEP's high-energy phosphate, making the transfer to ADP super efficient, producing ADP and pyruvate, the end product of glycolysis. This step, along with the PGK step, is called substrate-level phosphorylation, SLP, because ATP is made directly by transferring a phosphate group from a substrate, like 1,3-BPG or PEP, to ADP, not through the proton gradient like an oxidative phosphorylation. Why do these reactions happen twice per glucose? You nailed it, because fructose 1,6-bisphosphatase splits into two molecules.
Perfect.
D8.
Okay, and let's talk now a bit about NADH. So NADH is oxidized by the mitochondrial ETC, but the inner mitochondrial membrane is impermeable to NADH. Explain this. Explain it in relation to carriers in the membrane to transport it across, and why this is the case. Also, talk to me about electrons from NADH entering mitochondria via shuttles. And we're going to talk about G3P shuttle in the brain and muscle. Why specifically G3P in the brain and muscle, you have to tell us. And malate aspartate shuttle in the liver and the heart. And explain us the goal of regenerating NAD plus and making ATP. So yeah.
All right, let's dive into NADH and how it gets its electrons into the mitochondria, since the inner mitochondrial membrane is impermeable to NADH itself. The membrane's tight structure, designed to maintain the proton gradient for ATP production, blocks NADH from crossing directly. Instead, cells use shuttle systems to transport the electrons from NADH in the cytoplasm, from glycolysis, into the mitochondria for the electron transport chain, ETC, while also regenerating NAD+ to keep glycolysis running. NAD+ is crucial because glycolysis needs it to oxidize glyceraldehyde 3-phosphate, and without it, the whole pathway would stall. First, let's talk about the glyceraldehyde 3-phosphate G3P shuttle used in brain and muscle. This shuttle involves cytoplasmic NADH reducing dihydroxyacetone phosphate, DHAP, to glyceraldehyde 3-phosphate G3P via an enzyme called cytoplasmic glyceraldehyde 3-phosphate dehydrogenase. G3P then transfers its electrons to FAD, which is bound to a mitochondrial enzyme on the outer face of the inner membrane, forming FADH2. This FADH2 passes electrons into the ETC at complex 2, bypassing complex 1, which results in about 1.5 ATP per NADH because fewer protons are pumped. Why G3P in brain and muscle? These tissues need quick energy bursts, brain for rapid signaling, muscle for sudden movement, so the G3P shuttle is simpler and faster, even if it's less efficient in ATP yield. It matches their high-demand, rapid response metabolism. In the liver and heart, the malate aspartate shuttle is used instead. Here's how it works. Cytoplasmic NADH reduces oxaloacetate to malate in the cytosol using malate dehydrogenase. Malate, a small molecule, crosses the inner mitochondrial membrane via a specific transporter. Once inside, malate is oxidized back to oxaloacetate, regenerating NADH in the matrix, and those electrons go to complex 1 of the ETC, yielding about 2.5 ATP because more protons are pumped. Meanwhile, oxaloacetate is converted to aspartate, which shuttles back to the cytosol to keep the cycle going. Why this shuttle in liver and heart? These organs rely on sustained, efficient energy production, liver for metabolic processing, heart for constant pumping, so the malate aspartate shuttle maximizes ATP output, which fits their high-energy, endurance-based roles. The goal of both shuttles is twofold: get NADH's electrons into the ETC to drive ATP production via oxidative phosphorylation, and regenerate NAD+ in the cytosol so glycolysis can keep producing pyruvate and a bit of ATP. Without NAD+, glycolysis would stop.
Okay.
Cutting.
This is good stuff you're saying, but remember, dude, let's try to keep it a bit more short and concise. Okay, let's talk about the G3P shuttle a bit in the brain and muscle now. So we've got what? We've got in the cytosol DHAP becoming G3P. We've got oxidation of NADH to NAD plus in the cytosol. Then we have G3P getting into getting past the outer leaflet, and DHAP going outside the outer leaflet. And then we have G3P becoming DHAP and FAD becoming FADH2 with G3P dehydrogenase, and we have this electron transport chain in the inner leaflet. Now, I don't understand anything about this. Could you tell us 2-3 sentences about it?
The G3P shuttle in brain and muscle moves electrons from cytoplasmic NADH to the mitochondria. NADH reduces DHAP to G3P in the cytosol, regenerating NAD+ for glycolysis. Then, G3P donates electrons to FAD on the inner mitochondrial membrane via glyceraldehyde 3-phosphate dehydrogenase, forming FADH2, which feeds electrons into the electron transport chain at complex 2 to produce about 1.5 ATP. This shuttle is fast and suits the high-energy, quick response needs of brain and muscle.
And with the malate aspartate shuttle, how does it work? So we have malate becoming oxaloacetate, and this kind of causes a reduction of NAD plus. Fuck knows why. And then it becomes from oxaloacetate aspartate, and then from aspartate it goes outside, and then back to oxaloacetate malate outside, then comes back in, and we have again this semi-oxidation of NADH to NAD plus, and you have this loop, huh?
All right, let's break down the malate aspartate shuttle in the liver and heart, keeping it tight. Cytoplasmic NADH reduces oxaloacetate to malate in the cytosol, regenerating NAD plus for glycolysis. Malate crosses into the mitochondria, where it's oxidized back to oxaloacetate, producing NADH in the matrix that feeds the electron transport chain at complex 1, yielding about 2.5 ATP. Oxaloacetate is converted to aspartate, which shuttles back to the cytosol, becomes oxaloacetate again, and the cycle loops, keeping NAD plus available in the cytosol and electrons flowing for ATP production.
Okay. Let's talk about the role of pyruvate in metabolism. So it's got a kind of few stuff. So pyruvate is at a crossroads in metabolism. And so we can get alanine, acetyl-CoA, lactate, oxaloacetate as a result.
Pyruvate is indeed a metabolic crossroads, acting as a key hub after glycolysis. It can go four main ways depending on the cell's needs and conditions. First, under aerobic conditions, pyruvate enters the mitochondria and is converted to acetyl-CoA by pyruvate dehydrogenase, feeding into the citric acid cycle to produce energy. Second, in low oxygen conditions, like during intense exercise, pyruvate is reduced to lactate in the cytosol by lactate dehydrogenase, regenerating NAD+ to keep glycolysis going. Third, pyruvate can be transaminated to alanine in the cytosol, especially in muscle, to shuttle nitrogen for protein metabolism. Fourth, pyruvate can be carboxylated to oxaloacetate in the mitochondria by pyruvate carboxylase, supporting the citric acid cycle or gluconeogenesis. These paths let pyruvate balance energy production, oxygen availability, and biosynthetic needs.
And when does switch happen?
The switch between pyruvate's metabolic fates.
When does each situation happen?
All right, let's break down when pyruvate takes each path. Pyruvate goes to acetyl-CoA in the mitochondria when oxygen is plentiful, like during normal aerobic metabolism in most cells, to feed the citric acid cycle for maximum ATP production. It turns into lactate in the cytosol when oxygen is low, like during intense exercise in muscle or in red blood cells, to quickly regenerate NAD+ for glycolysis to keep running. Pyruvate becomes alanine in muscles, especially during exercise or fasting, when amino groups need to be shuttled to the liver for protein metabolism or gluconeogenesis. Finally, pyruvate converts to oxaloacetate in the mitochondria when the citric acid cycle needs replenishing or during gluconeogenesis, like in the liver during fasting, to make glucose. The switch hinges on oxygen levels, energy demands, and whether the cell prioritizes energy production or biosynthetic pathways.
So how do we choose? I don't understand. How will we choose how each situation occurs? Quiz us a bit. So also with the enzyme like lactate dehydrogenase and alanine, and pyruvate dehydrogenase complex 2 and pyruvate carboxylase. I want to guess which enzyme leads to which situation. And then I want us to talk a bit about the possible fate of pyruvate more. So when we have sufficient oxygen, the pyruvate can be oxidized to carbon dioxide and water and generate ATP. Okay, this I understand. So from glucose, we have glycolysis, we have pyruvate, then we have lactate, and we get, and so with pyruvate and oxygen, we go from pyruvate instead of lactate. Okay. So in hypoxic conditions, lactate is produced. Okay.
All right, let's clarify how pyruvate's fate is determined and which enzymes drive each path, keeping it concise. The cell doesn't really choose pyruvate's fate, depends on oxygen levels, energy needs, and specific enzyme activity triggered by those conditions. Let's break it down. Acetyl-CoA. When oxygen is sufficient, aerobic conditions, pyruvate enters the mitochondria and pyruvate dehydrogenase complex, PDC, converts it to acetyl-CoA. This feeds the citric acid cycle, leading to complete oxidation to CO2 and water, producing up to 30-32 ATP per glucose via oxidative phosphorylation. This happens in most cells, like in the liver or heart when oxygen's abundant. Lactate. In low oxygen, hypoxic conditions, like during intense exercise or in red blood cells, lactate dehydrogenase, LDH, reduces pyruvate to lactate in the cytosol. This regenerates NAD+ to keep glycolysis going, yielding only 2 ATP per glucose. It's a quick fix for energy when oxygen can't support the mitochondria. Alanine. When muscles need to shuttle nitrogen to the liver, like during fasting or intense exercise, alanine aminotransferase, ALT, converts pyruvate to alanine by adding an amino group. This happens in muscle to transport nitrogen for urea synthesis or gluconeogenesis in the liver. Oxaloacetate. In the liver, especially during fasting or when the citric acid cycle needs a boost, pyruvate carboxylase, PC, converts pyruvate to oxaloacetate in the mitochondria. This supports gluconeogenesis, making glucose, or replenishes cycle intermediates. For a bit more on pyruvate's fates, these paths are like a metabolic traffic hub. Oxygen availability is the main signal high. Oxygen pushes pyruvate toward acetyl-CoA for maximum energy. Low oxygen forces the lactate route for quick NAD+.
Okay, that's so good. So we have under aerobic conditions complete degradation of pyruvate in the mitochondria. Why specifically in the mitochondria? And I want to also ask you about transport of pyruvate into the mitochondrion. So we have a carrier protein in the mitochondrial membrane. Talk to us about that. And then we have pyruvate undergoing oxidative decarboxylation by the pyruvate dehydrogenase complex, and it forms acetyl-CoA. Explain why this is needed. So we have, So we have pyruvate plus CoA plus NAD plus going to acetyl-CoA, carbon dioxide, and this is an irreversible reaction. Explain why it's irreversible. Do everything of these things I asked in maybe 4 or 5 sentences. I know it's kind of a good challenge. Let's do it.
Pyruvate is fully degraded in the mitochondria because that's where the citric acid cycle and electron transport chain are, equipped with enzymes and membranes to maximize ATP production from pyruvate's oxidation. Pyruvate enters the mitochondria via a specific carrier protein, the mitochondrial pyruvate carrier, which selectively transports it across the inner membrane to reach the pyruvate dehydrogenase complex, PDC. The PDC catalyzes oxidative decarboxylation, converting pyruvate, CoA, and NAD+ into acetyl-CoA, CO2, and NADH, feeding the citric acid cycle to generate energy. This reaction is irreversible because the loss of CO2 and the high energy transfer to NADH and acetyl-CoA make the reverse process energetically unfavorable. The setup ensures efficient energy production and ties pyruvate's breakdown to the mitochondrial machinery.
Okay, let's talk a bit now about TCA cycle, also known as citric acid cycle, Krebs cycle. So this is a final common pathway for the oxidation. Explain why we say it's a final common pathway. And you have acetyl residues, they oxidize to carbon dioxide. So we have 4 redox reactions. We have a lot of involvement with NADH and FADH2. We have 1 ATP produced from each round. And I want you to explain to us these 8 intermediates. So we have citrate going to isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, L-malate, oxaloacetate, and eventually with acetyl-CoA we go to citrate. So in these stages we have NADH, we have FADH2, again NADH. What's going on? Guys, put a picture on Google of the TCA cycle.
So, we were just talking about FADH2 and ATP and metabolic pathways, and now we're diving into the citric acid cycle, also called the TCA or Krebs cycle, to cover those 8 intermediates and the production of NADH and FADH2. The cycle starts when acetyl-CoA combines with oxaloacetate to form citrate in the mitochondria. Citrate is rearranged to isocitrate, then oxidized to alpha-ketoglutarate, producing NADH from NAD. Alpha-ketoglutarate is further oxidized to succinyl-CoA, generating another NADH and releasing CO2. Succinyl-CoA is converted to succinate, producing 1 ATP via GTP in some cells through substrate-level phosphorylation. Succinate is oxidized to fumarate, forming FADH2 from FAD. Fumarate is hydrated to malate, which is then oxidized to oxaloacetate, producing a third NADH. This cycle runs twice per glucose because each glucose makes 2 pyruvates, hence 2 acetyl-CoA molecules, with each round yielding 3 NADH, 1 FADH2, and 1 ATP. These NADH and FADH2 feed the electron transport ATP production, while the cycle regenerates oxaloacetate to keep spinning. For visuals, searching TCA cycle diagram on Google will show you the full loop with all intermediates clearly laid out. Want me to quiz you on the steps, or focus on something specific in the cycle?
Well, I'd be really bad with quizzing the steps. We have a lot of enzymatic steps too, like we haven't mentioned, like axonitase and succinyl-CoA synthetase, succinate dehydrogenase. And I want to keep this episode on 30 minutes. So perhaps if you could do a nice kind of explanation of the enzymatic steps in a couple of sentences, which is, impossible. And then I want us to talk a bit about the regulatory points of the TCA cycle. So which are the reactions that are irreversible and the main regulatory points?
Alright, let's wrap up the TCA cycle in a tight package to keep this under 30 minutes. The citric acid cycle has 8 enzymatic steps. Citrate synthase combines acetyl-CoA and oxaloacetate to form citrate. Akinetase rearranges citrate to isocitrate. Isocitrate dehydrogenase oxidizes isocitrate to alpha-ketoglutarate, making NADH. Alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate to succinyl-CoA, producing NADH and CO2. Succinyl-CoA synthetase transforms succinyl-CoA to succinate, generating ATP or GTP. Succinate dehydrogenase oxidizes succinate to fumarate, yielding FADH2. Fumarase hydrates fumarate to malate, and malate dehydrogenase oxidizes malate to oxaloacetate,
Okay.
producing...
Let's talk about the products now. So we have 3 NADH, 1 FADH2, 1 GTP. Why is it called GTP and not ATP? 2 carbon dioxides. And let's talk a bit about the components of the TCA cycle. So we have, as intermediates. So what does it mean to be replenished by anaplerotic reactions? And
Alright, let's dive into the products of the TCA cycle and your questions about GTP and anaplerotic reactions, keeping it concise for the podcast. Each round of the TCA cycle, starting from 1 acetyl-CoA, produces 3 NADH, 1 FADH2, 1 GTP, and 2 CO2 molecules. Why GTP instead of ATP? The enzyme succinyl-CoA synthesase transfers a phosphate from succinyl-CoA to GTP, forming GTP, which is energetically equivalent to ATP and can be converted to it by nucleoside diphosphate kinase. It's just a quirk of the enzyme specificity in the cycle. The two CO2 molecules come from the decarboxylation steps at isocitrate to alpha-ketoglutarate and alpha-ketoglutarate to succinyl-CoA, releasing carbon from pyruvate's breakdown. As for the intermediates, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. They can be depleted when used for biosynthesis, like making amino acids or glucose. Anaplerotic reactions replenish these intermediates. For example, pyruvate carboxylase converts pyruvate to oxaloacetate, or amino acids like glutamate can feed into alpha-ketoglutarate, ensuring the cycle keeps running without stalling. This keeps the TCA cycle spinning to produce energy and support other metabolic pathways. Want to wrap up with anything else before we close
That
out
was perfect. Thank you so much for watching part 2, guys, and see you next time.